Pavel Tomančák is a renowned evolutionary and developmental biologist who had been leading his own research group at the prestigious Max Planck Institute in Dresden for 15 years. This year, he became the new director of the CEITEC Consortium. He will introduce us into how practicing of open science impacted his own career. So, Professor, the stage is yours.
Can you hear me?
Yes, we can.
Okay, great. So first of all, I'm not a professor, but that's great. Thank you very much. I appreciate that in Czech Republic, I almost invariably become a professor. I have been kind of expecting this to be actually in-person event, so I have to kind of a little bit adjust my presentation style to this. But basically, the idea was to introduce some kind of a practical view on how open science can, in principle, positively affect not only scientific community in general, but one's own, let's say, scientific career. And I would like to maybe just preamble this by saying, this is my personal, let's say, story, my personal journey. Of course, some of the choices which I have made, may not work out always in such a positive way as I will probably present it. Nevertheless, it's an example of how it can work, and I think in the current, let's say, scientific climate, it's in fact becoming more and more safe to practice open science, and this is because these ideas, which I will discuss a little bit, actually are now adopted by the mainstream, and the major funding organizations around the world, and especially in Europe, in the European Union, are really mandating. There's some background noise, by the way. Are mandating open science, or open access to scientific data and other aspects of science. So in principle, now basically it's almost required, and one can say that that's good, and it's a result of essentially of the revolution which has been started towards open science within the scientific community, and has been now adopted by the funders and by the, let's say, leadership of the scientific community. But it's a very kind of bottom-up approach, I would say, because scientists realize that it's extremely important to be open, and many scientists have been open even before open access or open science even existed as a term. Okay, so that's the preamble to what I was talking about. I would like to basically give you a few examples. I'm trying to get some information about what time is it, because I don't know, I don't have a watch. But anyway, a few examples of how one can be open in science, and this will be in 4 different areas. It will concern data, software, hardware, and at the end also ideas. It is very rooted in my research. I've been presented as an evolutionary developmental biologist. I don't know what the audience is, but my guess is that majority of the people will be more oriented on the technical sciences. So I will try to somehow, keep the jargon of biology as low as possible, but I cannot avoid it because my examples are really about biology. So hopefully you can get something out of that. So I will start basically by saying, that for me, the journey in open science started already in my PhD, where around, let's say, the year 1998, I was studying a specific gene, which is called Paraparvan, but that is not important, which is essential for establishing the body axis of 2 different animals. It was known that for the C. elegans worm on the left side, this protein is localized on one side of the one-cell embryo. And I found during my PhD that the same protein, equivalent, we say biologists say homologous protein, is also localized at the one end, we call it a posterior end of the oocyte, which gives rise to a fruit fly, right? So that was, in fact, quite amazing that this kind of one molecule seems to be extremely important for establishing what is the head and the tail of a worm and a fly. In the process of studying this gene, I needed to clone that gene. And this turned out to be extremely difficult because the gene was complex, right? So here is shown the structure of such a gene. Of course, you would have to zoom in to see the actual sequence. There would be thousands of nucleotides which form the genetic information, the DNA. But we usually show it in the kind of schematic view, how the gene is kind of built together from the pieces of genetic information. And this is what I've eventually found out, how complex this is. And this, in order to get there, one needed to really do, back then in 1998, a lot of work of sequencing the DNA, which, would have taken me, using the techniques which were available at the time, probably many years, and I would probably not even, finish in time for my PhD. And so that's why for me it was completely kind of transformational that around the time there was a publicly funded genome project which was aiming at sequencing the entire genome, the entire genetic information of a fruit fly. They did that with a very targeted approach. So they were focusing on different parts of the genome and sequencing it, using the public money. This was happening in the United States, in the laboratory of this gentleman, Jerry Rubin, very famous geneticist. And so they happened to actually, in the early stages of that project, focus on the region where my gene was. And so then my task basically has become to go online and download the sequences which they actually generated, right? So this was really transformational. It helped me very much because I could then piece together this structure very, very quickly, and had a massive impact on my scientific career, I must say. So I actually was so enchanted by this openness, because they basically put the sequences online the moment they assembled them. The moment they came out of the sequencing machine, they were put online and you could use them. I was so enchanted by this that I joined the laboratory of Jerry Rubin, and I started somehow then practicing open science myself together with let's say, him. And we encountered another project, which is going to be a little bit difficult to explain, but nevertheless, at that time, there were very specific new kind of genes, which were not making proteins, but which were, purely RNA. They were really small. That's why they were called microRNAs. And they were a novel phenomenon. And so basically they were somewhere in the genomes, but we didn't really know where, right? And so we, together with a postdoc in Jerry Rubin's lab, with Eric Lai, we devised the computational scheme how to find those genes in the genomes. And what we used really kind of as an approach here was that we needed to find out, we had two clues of information. One was that these genes, they have a specific structure. They create this kind of, what is marked here on the slide as a conserved stem loop. They have a certain structure. When you put them into a program which folds RNA, they create this kind of structure. And because they are genes, they do something, they are conserved. So two different species, Drosophila melanogaster, which is the fruit fly everybody studies, and we have a complete genome of it, and another fruit fly, Drosophila pseudoobscura, they will have the same genes, almost guaranteed, because they are very related to one another. And so at that time, another genome project was starting to sequence the genome of this related fly, Drosophila pseudoobscura, and they were also putting the data out in the public domain. And so what we were able to do with Eric was to collect those sequences, so-called align the genomes, finding which part of the genetic information is similar, which is shown here by these red peaks. And then we would pass them through this folding program using a computer cluster and ask, which one of these conserved peaks, which have the same sequence across those two species, which ones form these loops and therefore are likely microRNA genes. And we were able to find all the microRNA genes in the Drosophila genome. So here, once again, the openness of the genomics community, basically sequencing and putting the data immediately into the public domain, allowed us to really generate profound insight into the structure of the genomes by finding the microRNA genes at the very early days when this was when this research field just about emerged, because those genes were just discovered. This something project like this impacts your scientific career enormously, because the next 10 years, the biology community, especially the RNA community, has been studying microRNAs. And everybody who publishes a paper about microRNAs cites you because you found the genes, right? And so this is actually my highest cited pure biology paper, because people really like it. And it was enabled by the openness of sharing data across the genomics community. Okay, so then I was essentially, by these two examples, sold to the being open about data, and I used it further to, let's say, disseminate the results of my own research. And this is something which has been kind of spanning across the work I did in the US at Berkeley with Jerry Rubin, and what I started then to do when I moved and established my independent research group at Max Planck in Dresden. So what we were doing, we were collecting images, two-dimensional images or maybe three-dimensional images of so-called expression patterns. So what this, in order to understand that, if you are not a biologist, is that genes in genomes, they are expressed in certain specific cells of the animal or of a living system. They are not expressed everywhere. Some are, but many are not. They are expressed in a specific tissue or are expressed in a specific time. And in order to describe that, you describe the pattern of expression. And you can do that by various means. Some of them are very high throughput, but what really is really telling is to photograph the living system and visualize where a particular gene is expressed. And so that's what we did very systematically for many genes in the genomes of the model species which we studied, which was Drosophila. And we assembled really large databases, hundreds of thousands of images, which describe the expression patterns when and where genes are expressed in different tissues of the fly. What you see here is the data sets on embryos with two different techniques which provide different resolution, and then additional tissues within the fly. Right? Those are resources which are incredibly useful to the scientific community. They are used to this day, even though the technologies have maybe surpassed them, because we have now different ways how to visualize gene expression patterns. Nevertheless, back then, this was around 2000, we were actually collecting this data, and like the genomics community, we were putting them right away onto a website so that people can use them. And similarly to what happened with the genomes, I mean, the scientific community did use the data. Eventually, we published the paper, and that paper is also very highly cited, because anyone who is looking for an interesting gene to study usually starts from these databases looking, whether this gene is expressed in a particular tissue, or if you do some kind of more global analysis where you are asking, questions about many different genes, you can ask, what are the types of tissues these genes are expressed in. So this is very much used, and I think having this kind of data sets out there in the open is completely crucial. And of course, nowadays everybody does that. But back then, we were quite pioneering in that we were putting the data from the microscopes directly online. Okay, so I actually continued doing that. This, in fact, to kind of demonstrate how it affected my scientific career, this type of project definitely landed me the job in Max Planck in Dresden. So for me personally, being open and sharing my data was really beneficial, because I could become independent researcher and a very nice prestigious institution, and back in Europe, which was also nice, but that has nothing to do with open access. Okay, so I continued at Max Planck to kind of bring this sharing to yet another level. So as I said, we have different ways how to visualize the patterns. Living systems are dynamic. I hope that this video somehow shows that. And what you are looking at here is also a gene expression pattern, but this time we can look at it live as the animal develops, right? So this is an embryo of Drosophila. We are imaging it with a very, advanced microscopy technique. And one gene is marked, tagged with a fluorescent protein, which we can see. And so we can see, how the expression pattern develops across time, right? Now we have to realize that this is not just image, right? There is a really transgenic fly, a fly which carries this marked gene, which you have to maintain and which you have to have in order to collect such a video. And so what we did, we also shared those resources. So we shared the sort of molecular biology reagents, but in particular, what is important here, the 880 fly transgenic lines, we shared them with the scientific community, and they are used to this day, right? This project is already about 10 years old. Once again, technology moved forward. This is surpassed by other projects which are doing it in a different way, maybe more targeted. But nevertheless, these 880 transgenic lines are a great resource to the, especially Drosophila research community, because people can order these flies and they get a gene shining, specifically a transgenic fly which has a specific gene marked, which one can image and one can do other genetic manipulations with or biochemistry. So this for me is, let's say, the end of data sharing examples. It's the ultimate, let's say, level where you really generate the transgenic animal and you share it across the community. And one has to say this is absolute standard now in the community, not only in Drosophila, but in mice, in any organism. When the work is put into generating such a transgenic animal, it is shared with anyone who wants to use it. It's safe, and it is definitely good for the scientific community and for your career. Okay, so now we will move to software. This is a long story, which I will have to try to make a little bit shorter. If you are a biologist, the next slide, you will know what I'm talking about. If you are not, I have to explain it a little bit. There is an open source platform for research in biological image analysis, which is called Fiji. It is a self-referential acronym. It stands for Fiji is just ImageJ. I will have a little bit more to say about that. And believe it or not, this is at the moment the dominant platform for analyzing biological image data. I will show you some numbers to demonstrate that. If we would be in one room, I would ask who uses Fiji, and if it would be a predominantly biology audience, 90% of the people would probably raise their hands, right? One has to say that in 2008, Fiji didn't exist at all, in fact. Right? So I was one of the people who stood at the birth of Fiji. And over the 10 years, it has become really something like, I compare it to Internet Explorer from Microscope in the web browser, right? There are other browsers which are, heavily trying to displace it, but it's very, very hard. For Microsoft Explorer, maybe it worked out, then for Mozilla and so on, and Chrome, but one has to really realize that if you achieve dominance in the software field, in particular here, we are talking about a massive field because everybody in biology collects images and everybody needs to analyze them. So Fiji is the dominant platform where people do that. It comes very much from history, which I don't have really that much time to work on because Fiji is just ImageJ and it builds on ImageJ. And ImageJ is what biologists have been using for many, many decades, right? But this is a story about Fiji. Okay, so why is Fiji so popular in the biological sciences? It is because, first of all, it's building on ImageJ, so it provides a familiar user interface. It has been, back in the days when it was starting, very well documented. There was a very active wiki where one could really learn anything about it. What I think made Fiji really successful also was that it was sort of not really providing the biologists with just the solutions, but it was also inviting biologists to extend it, right? So Fiji and ImageJ have this plugin architecture which allows anyone to use the platform to add a specific solution to it. And this could be done in Java, which is quite involved, or it can be done using scripting languages, which many biologists, handle extremely well. And so this kind of opened the possibility for biologists or even nowadays people would call somebody who specializes in writing software for data analysis, biological data analysis, bioimage analysts, to create their own solutions for their specific problems very, very easily. What made Fiji, what distinguishes Fiji mainly from the classical vanilla ImageJ developed by Wayne Rasband, a very important name to mention here. That's really the true father of Fiji and of ImageJ and its offspring Fiji. So what Fiji distinguishes itself is that it invested heavily in software engineering, which means that it provided people who are professional at developing software with the tools of the industry which they are used to. Right? They are always, you know, I'm sitting here at CEITEC, and next to me are 3 buildings full of programmers for Red Hat. Right? It's very, very difficult to get programming talent into open source project such as Fiji when they can work at Red Hat, right? And it would definitely not work if they are not provided with the tools they are used to use. I mean, those are all acronyms which, you know, in the biology community are actually, you know, deeply hated because nobody really knows so well how to use them. But the professional software engineers, they want to be in an environment which works like this, and that's what Fiji provides them. And lastly, what on the bottom of the slide, what we really wanted to do, we wanted to bring into biology new ideas from computer vision, and we wanted to attract researchers in computer science to really, you know, take their newest algorithms and transfer them into programs which can be applied to biological images. And for that, we developed libraries which make it really relatively easy to do. And those are the ones which are on the bottom side of the slide. They are very powerful. They really scale to large data, but they're not super easy to use and to learn. Okay, so now very often people ask what is the difference between Fiji and ImageJ, and one could really kind of explain it very graphically, so that ImageJ is the core, is the platform on which Fiji builds this kind of application layer. I have a little joke here, which actually demonstrates that even though my current involvement with Fiji is maybe a little bit diminishing, I will always remain the founding father of Fiji because my initials, will it work? Because my initials are actually engraved in the Fiji logo. If you take the inverse of it, you get a PT, which stands for Pavel Tomančák, obviously. I have to say this is, I mean, if we would be in one room, we would probably, or hopefully at least, laugh a little. One would say, you know, was that by design? Absolutely not. I learned about that maybe 2-3 years ago. I didn't really see it myself, but somebody pointed it out to me. So it's a complete coincidence, but it's there in the logo and nobody can remove it anymore. Okay, so what Fiji is about is solutions for big image data. There's some examples here, which really is pointless to try to explain them, but just to say, you know, nowadays biologists are peering into the incredible complexity of biological systems with very powerful automated microscopes. In fact, you know, we could say robotic microscopes. We are not really, you know, looking through the binoculars. The microscopes are essentially autonomous, and they collect, you know, terabytes of image data, really incredibly complex, large data sets. And, you know, at this data, one cannot even look at them anymore because they are too big. I mean, 100 terabytes of image data, you can spend your entire life looking through it. So in order to put them together and to analyze them, you need computers and you need software, which is able to really sift through that data, which is able to reconstruct it, which is shown mostly on this slide, and also to analyze it, right? And here is where Fiji essentially comes. It is a platform which really aspires to develop the state-of-the-art tools to analyze the massive data sets which are coming out of biology these days. Okay, this actually is not animated, but nevertheless, you know, I said that Fiji is used everywhere. We have a very precise usage as statistics. The number of users can be estimated between 75 to 100,000 on a regular basis. And, you know, we can really demonstrate that it is being used by the fact that the paper which we published about Fiji in 2012, now has 31,894 citations as of today, right? It is being cited now at the rate of about 8,000 citations a year, right? So that is indicating that people are really using it. Right? It will not stay like that. There will be other platforms which will be used, which will be better because they will be building on the green field, and they will not be having to deal with the legacy issues which Fiji has. But nevertheless, you know, this is really indication that this is used everywhere. What has really been remarkable for me, and now I come back again to the topic of open science. So of course, you know, Fiji, ImageJ, everything is open source. You can look at every piece of code. If something is not open source, it will not be in Fiji. It is essentially not allowed. Right? We have a certain licenses which we do allow, and they are all open source. They have various flavors. Nevertheless, you must be able to examine the code, right? And so what really has been, you know, incredibly beneficial for me and for everybody involved is that these open source projects, they create a community of people. We have been organizing, you know, since 2008, the so-called hackathons, where we essentially invite the Fiji developers to one place. Initially, it was usually Dresden, but then later on, you know, many other places. And we lock them in the room, provide an ample supply of coffee and fast internet access, and they have their own computers, and they work together. Usually do this for 1 or 2 weeks. There's incredible so-called hackathon effect after that. People have the ability to talk to each other. They program in pairs. The amount of expertise in the room is so incredible that people make giant leaps on their projects where they are often stuck. And these hackathon effects, they usually last much longer after the hackathon, where people somehow go back home and they finish up the stuff they started at the hackathon. This is a really historical picture of one of the very first hackathons in 2008. By the way, most of the people here in the room, sadly not all, but doesn't always work out that way, now have, you know, very, very good jobs in academia or in industry, usually leadership jobs. The hackathons now look more like this. This is now fast forwarding 2017. This is one hackathon also done in Dresden. And this is how many people we sometimes attract, something like 50 people. It's not only about Fiji, there are other open source platforms for biological image analysis which are coming, and we are really investing in building bridges between them. But for the purpose of this talk, is just want to show that it creates this, you know, incredible community of people who really know each other, who like to work with each other, who like to meet, and who really are, you know, providing essential service to the scientific community by developing this, you know, very complex and very powerful software tools which everyone is then allowed to use. And this is all thanks to open science and open source. One of the really fantastic recent developments have been also the ImageSC Forums. So it's very, you know, people have always have questions about the software, have suggestions. Many things don't work, bugs need to be squashed. This is really the dynamics of the open source community, which is sometimes very unpredictable. And so what my colleagues from Madison and Ann Carpenter have been able to do is to put together, to unite, you know, all these platforms which are doing open source software development in this arena around microscopy, into these ImageSC Forums, which are incredibly active. That this is the places where the developers and the users meet, and you can post the question there. And if it's not answered, it will hang there, and it will kind of, you know, work in the back of the mind of the developer that he should answer this at some point. And really, this is a fantastic place how to solve your problems and how to push these platforms forward by the productive interaction between the user and developer community. You know, this is something which really will change this field forever. Now, these days, there are many initiatives which really support open science programmatically. And one of the most prominent one among them is the Chan Zuckerberg initiative, which really, you know, puts the, you know, tremendous money available to its founders to support open source software development. In fact, today, this afternoon, this evening, I will for the first time be present at the meeting of the Chan Zuckerberg Initiative, which is really supporting many open source software projects, including Fiji. It so happened that we were actually, it was somehow, for our project beneficial to bring the Chan Zuckerberg Initiative grant to Brno, to CEITEC, right? So now, in fact, even though CEITEC was not really known for being a center of Fiji development, in fact, it will become one because we have funding to hire some programmers to do software development in Fiji, especially with respect to these big challenges which I showed, the big data, the terabytes of image data, and the ability to really analyze and navigate them. We are bringing that to CEITEC. So, which leads me to my little advertisement. We are looking for the people who want to join this amazing scientific community, who want to help scientists around the world, who want to become part of the Chan Zuckerberg Initiative family, and work on the most, in my opinion, the most exciting problems one can work on to uncover the secrets of life with big computers. Okay, so that also means that you could join, you could work with these three gentlemen who, have a lot in common. Their brains, are incredible, and they are incredibly talented. We can see another thing which they have in common, but I will not comment on it. It is something which I don't have in common with them, let's say. And, I want to show it here just to demonstrate, how openness has been useful for them, right? So the ones, the Stephans, they did a PhD with me, and now they are both leading their own extremely successful high impact research groups in the United States at the Janaria Research Campus, which is the mecca of this kind of big biology data analysis. And they are there, and they are shaping and forming it. And Tobias Peach is a freelancer now. He has a little company where he, basically makes money by providing his incredible knowledge about the infrastructure which is behind Fiji and the big data analysis framework which is associated with it. So for them, it worked out incredibly well. I think that if they would not be participating in this kind of open source, open science initiative, their careers would not be as successful as they are. For me, it is, as I will repeat again, it is an opportunity to really work with these fantastic people. For example, once we assembled a very large Fiji logo in the atrium of the conference center in Heidelberg at EMBL, which is the mecca of molecular biology across Europe. And we had a serious discussions about whether to leave it there permanently, because Fiji is something which they use a lot there. But at the end, it had to be dismantled. So that was very nice. And this community is really active. Even COVID couldn't stop us. We were organizing relatively recently a really big event at Janelia, where there were something like, I think, 40 tutorials on various aspects of biological image analysis. 800 people registered. It was incredible. And we were, one of the most active community using the Gathertown to actually socialize during the conference. The conference was going on across all time zones, across affecting basically people worldwide. And we had incredible fun on this little island which we developed. And being geeks, we even played some games, some treasure hunts, and being computer geeks, people have really brought this to another level, basically hijacking some kind of HTTP requests and building robots of the little avatars in this virtual reality environment, which were scanning the island for the hidden doors. And this so-called Blair bot actually solved the game purely automatically as a remote controlled robot, which we found very, very impressive. This is just to whet your appetite that this is really incredibly talented community of people. Okay, so now we will move to almost the last part. I think I have 45 minutes. If not, then stop me. My understanding is that I'm 30 minutes in. Okay, so now hardware, right? So that's getting a little bit actually unusual. Everybody knows about open source software. Now, what about hardware? So once again, this is a personal story. There are many open hardware projects which I don't have time to talk about. The one which I got interested in was the one which is related with the SPIM microscopy. SPIM stands for Selective Plane Illumination Microscopy. Once again, this is better for biology audience. Okay. But we started, now I got a message that it's 10 and the next presentation should start, but we started 15 minutes later, right? So that's gonna be maybe a little bit difficult, but okay, so I can speed up, all right. So this is a microscope, which for the non-biology audience, maybe is no point of explaining how it works, but it really allows you to generate incredible insights into living systems by imaging them for a long time without damaging them and imaging them in 3D. I will show you one example of that. Just play the video. Here you can see a biological system, a shrimp, which is developing. We can see every cell imaged with this microscope. It is sped up many thousand times. And now as we are zooming in, we see really the spectacle of life happening where it creates forms. The shrimp has a different appendage, different body appendage on every segment, and we can see them formed here, right? So these microscopes allow you to create such data. This shrimp is imaged for 4.5 days, and it creates really big data sets. So every biologist would like to use this kind of microscopes, but back then, maybe 10 years ago, it was a bit difficult because these microscopes, they were only existing at the technology development stage, at the optical tables of the technology developers. And it took quite some time before they were commercially available. So what we contributed to this was to bring this technology closer to the users early on, kind of early access, by developing a platform which is called OpenSPIM, which is a DIY, which stands for Do-It-Yourself Light Sheet Microscope. So what is it? It's a very simple light sheet microscope, which you can build yourself. There is a wiki which will tell you exactly how to do it. There will be a list of parts which you can all buy, or some of those parts you can get them ordered, made, machined, or you can 3D print them. There are cut drawings which allow you to do that. Then there are these texts and videos which show you really how to put it together down to every single screw. These videos are narrated by an American person who has been behind this project, Pete Petron. And so, according to this video, even if you have never ever built a microscope, I guarantee you can do it. In fact, it is relatively simple to assemble such a microscope. There are essentially 19 steps. And the added value of this open access project is that we introduced the assembly instruction, which are on the Lego IKEA style, right? So anybody who, has a screwdriver, an ability to buy the parts, which actually are not cheap, right? I mean, you still end up with 30,000 euros worth of parts to assemble this microscope, you can actually build it. So it's a little bit like a BMW. It's still expensive, but these microscopes are expensive. Okay, so then you get a microscope which you can put in a suitcase, and it can generate really nice data. Once again, for non-biologists, this will not mean much, but here we are looking at the scan of a fish, and here we are looking at the time-lapse development of Drosophila. It's of course not the best microscope on the market. It's not the fastest. It's not the easiest to use, but you can do it yourself. So it's a fantastic tool to teach and disseminate. We have been going with this microscope to many different courses, also in South Africa. And so it is a tool where you can reach with this kind of fascination of biology and high-end microscopy, even to high school students in places which usually don't get access to this kind of high-end equipment. The OpenSPIM is a simple microscope, but you can make it more complicated. You can develop all kinds of designs which are bringing in different illumination detection axes. And because it's so simple, you can also multiplex it. You can build several of those and you can image several specimens at the same time. Those were concepts which we put out in the paper, which we published about this. But in fact, you know, they were almost all of those were implemented once again by the community, because by being open, we have kind of sparked a community of people who are building these open SPIM microscopes. In fact, we know that there is about 150 of those built around the world. They are not everybody is completely satisfied with it. Not everybody is building exactly what we built. Many people build something very different. These microscopes can sometimes be not even recognizable as open SPIM. Nevertheless, I'm surprised, you know, as I travel around the world before COVID, that you enter a lab and you see, okay, this used to be an open SPIM. So this is really great in terms of, you know, sparking also the possibility for the biology community to participate in the development of the hardware. For me personally, it has been really beneficial because I was able to actually make somebody who studies these kind of animals, flatworms, to build a microscope and to publish a paper about his experience of building it, right? And then I actually hired that person as a postdoc. And I find this, you know, extremely liberating that with the open hardware project, you can take a biologist, you can have a biologist who has never really programmed anything or never really built anything, and build a microscope which allows you to do the high-end science which you want to do. And that's the benefit here. I have one more slide, which is about ideas, which I will go through very quickly. So this is a little bit kind of philosophical. I mean, the question is, you know, how much can one afford to be open about ideas? I have been kind of, let's say, once again, personally practicing this quite a lot on Twitter, where that's a good place how to essentially, you know, bounce your ideas against other people. I think that the feedback which you get, you know, which can be sometimes not what you want to hear exactly, or can start some kind of a controversy or flame war, but if you use it right, you know, it's very good to kind of bounce your ideas against the community on scientific Twitter. What really is the driving force in this kind of open science area is open access publishing. Here I am really into it, but open access publishing is now become really expensive, and so, you know, one has to really, you know, be careful that this does not become, as Petr Svoboda says, a cargo cult, something that we do it because we are expected to do it. We are expected to do open access, right? We are mandated even, right? I think that it is much more important to be open about your science and about your data, your software, your hardware, and your ideas de facto than de jure, right? If you follow the rules, right? I mean, we have been very often, people pointed out to us that it's ridiculous that we publish something like open SPIM in a journal which is closed access. But to that, I argue you can get to the PDF anytime. The journal which did that did a fantastic job in putting the paper together. And the project is open access because you have a website where everything is basically there. So let's not be demagogic about it. I know that this will be controversial to say in the public video, but nevertheless, I want to say it. Finally, I think what really fuels, which becomes maybe even more important than the open access publishing, is the possibility to share your results in a form of preprints. This is something which I very much am into. I recommend it. I know that in Czech Republic, this is relatively in its infancy, but we need to catch up, right? Because there has been, especially in the technical sciences and in physics, there has been an archive forever. And it's common to share your results immediately with your colleagues in the scientific community in the form of a preprint. And biology is coming really hard, right? And essentially nowadays, you know, most biologists are preprint archiving their work. We don't have time to discuss what are the pros and cons of that, but from me, just here endorsement for this, because from my personal experience, preprint archiving has been, you know, fantastic way how to get the data out before the inevitable delay of publishing makes them essentially old. Okay, so that's basically my last slide where, you know, I managed to keep on time, even though I'm sorry we started a little bit later. I want to come up with the take-home message, which is essentially, you know, coming back maybe a little bit to the beginning, right? Now, open science is a big topic, right? I have been recently writing a grant, and there was in fact mandated that half a page is actually dedicated to discussing open science, right? And you know, and you have a lot of different mechanisms which we will probably hear about in the next from the next speakers, the fair principles, and you know, what was mentioned already, you know, to be as open as possible and as close as necessary, and all these kind of let's say mandates which the scientific community has actually arrived on, which is very, very good, right? But I think that we should not forget, you know, that fundamentally open science is about the mindset of the scientists and about their ability to see that by being open, you know, they get something out of it. And so my take-home message is that share your results of your research, share them early, share them often. Good things will happen to you.